Hello, class. Welcome to our lesson today. And in this lesson, we're going to be looking at cell division. So remember, this is a continuation from the topic that you were introduced to last time, which is genetics. So last time you looked at the terms used in the study of genetics. You also looked at variations. So this time we're going to commence our discussion of genetics by looking at cell division. So what are we expected to know by the end of the lesson? So by the end of our lesson, we should be able to describe the stages of cell division. We should also be able to explain the importance of mitosis and meiosis. So before we even delve into discussing the different forms of cell division and the like, I would like to pose a question. What would you say cell division is? Or what is cell division? What comes to your mind when you think of cell division? So to answer that, we can say that cell division is basically the multiplication of cells. We're looking at a process where cells are increasing in numbers. That is what cell division is about. So we generally have two types of cell division, and that is mitosis and meiosis. Those are the two types of cell division that we have and will be studied in this lesson. So to better understand cell division, we need to understand the components that are involved in this cell division. It will help us better understand what is basically happening in cell division. And one of the components that we need to understand that is involved in cell division is a chromosome. So what is a chromosome? And the time that you are discussing the terms that are used in the study of genetics, you came across a chromosome, and you most likely would have defined it as a thread-like structure of genetic material found in the nucleus. So what then do we mean by this? We are aware that the genetic material that we have in the nucleus is DNA. But how does this DNA, or how is this DNA stored, or in what form do we have it? So when you look at the chromosomes, our chromosomes are saying are thread-like structures of genetic material. Our genetic material is DNA. So when you look at the chromosome is basically DNA wrapped around proteins, allowing for it to easily be stored in the nucleus. We know that DNA, which is our deoxyribonucleic acid, contains genetic information, this information that we have inherited. So this inherited information, this genetic information occurs in the form of what are referred to as genes, okay? I hope we're clear to that. So when you look at our chromosomes, we're looking at thread-like structures of DNA that we find in the nucleus, okay? So these chromosomes allow for us to package this genetic material in the nucleus. So before cell division can take place, we have DNA getting replicated, meaning that DNA is making copies of itself. So during this replication process, we have DNA doubling in number. So to prepare for cell division, DNA needs to double in number. So as it doubles in number, it forms a structure of a chromosome that looks like an X. So it forms a chromosome that has got two strands, like we can see here. This is our chromosome. So it has got a strand there. It also has another strand on the other side. So this is in preparation for cell division, implying that when cell division is not taking place, if you were to look at the chromosomes, they wouldn't generally look like this. They would more of look like that, just a single strand, okay? But as cell division is about to take place, a chromosome is going to develop another strand, an identical strand. So it's going to have two strands in order to prepare for cell division. I hope we are clear to that point, okay? So our chromosomes, we've said, thread-like structures of DNA that we find in the nucleus. So when we look at our chromosome, like we mentioned, it has got two strands. So the two strands of our chromosome are what we refer to as sister chromatids, okay? So for every organism, there's a certain number of chromosomes that we have in the nucleus. So I'll be talking about us humans. So for us humans, our somatic cells or our non-sex cells, our general body cells, have got 23 pairs of chromosomes, implying that we have a total of 46 chromosomes that is in our non-sex cells. So in cells that are not gametes, we have 46 chromosomes, which is giving us 23 pairs, okay? So a pair we know is 2, right, isn't it? So implying that when you look at the chromosomes that are in our cells, they occur in 2s. So these pairs are what we refer to as homologous chromosomes. So you could think of it like every chromosome has got a best friend somewhere that it will be with in the cell. That is its homologous pair. So the two are referred to as homologous chromosomes. So in the previous slide, I mentioned how when a cell is not dividing, you could think of the chromosomes as being more single strands. But as it prepares to divide, it develops another strand, like we can see here. I like how this is, it's even being colored differently. So you have one strand and you have another strand there. So the two strands of the chromosome are what we refer to as sister chromatids. So our chromosome is made of two sister chromatids. The sister chromatids are joined to each other by a structure. I have over there, let me highlight it better. You have this structure here. This is the centromere. So two sister chromatids are joined to each other by a centromere, okay? Then when you look at the chromosomes in our body cells, they occur in pairs, and these pairs being referred to as homologous chromosomes. I hope that is clear to this point. So like I mentioned, our chromosomes before cell division would generally occur more like a single strand. Then as they prepare for cell division, because we have that replication of DNA, so you're going to have another strand. So it will be double-stranded. At the end of our cell division, we expect that the two strands will separate from each other. This is for stability purposes, so that the organism that is formed from this division is going to be genetically stable. Okay, we proceed. So we mentioned that we have two types of cell division. So the first type that we look at is mitosis. What is mitosis and what are the stages involved? When we look at mitosis, we basically look at a type of cell division where one cell divides to form two daughter cells that are genetically identical to it. I hope we've gotten that down. So in mitosis, a parent cell is going to divide and is going to form two daughter cells that are genetically identical, okay? So you have one cell, the cell is going through division. When it finishes its division, it ends up with two daughter cells. That is what mitosis is, okay? So for mitosis to occur, there are certain stages that will take place. So in the follow-up slides, we're going to look at these stages that are involved in mitosis. So the stages of mitosis are prophase, metaphase, anaphase, and telophase. So you can use the acronym PMAT to remind yourself, and it's in that order. So first stage is prophase, then we have metaphase, then we have anaphase, then lastly we have telophase. So let's look at these stages individually. Our first stage is the prophase. What happens during prophase? So in prophase, we have movement of centrioles to opposite poles of the cell. Centrioles, if you remember your cell structure, are part of the cell structures that produce other structures known as the microtubules, which are needed for cell division. So we have our centrioles. So our first stage was saying that centrioles, which are these, are migrating to opposite poles. So it's moving that side, even this other one is moving the other side. So they migrate to opposite poles. Then we have the appearance of what are referred to as spindle fibers from the centrioles. You can see on the centrioles that these structures that look like they're sticking out. These are the spindle fibers, so they're also going to appear. Then we are going to see the nuclear membrane break down, disintegrate, or disappear. So remember, the nucleus has got a membrane around it. So this membrane around it is going to break down. So if the membrane around the nucleus breaks down or disappears, it means that the chromosomes are now free to move within the cell. Remember, I said the chromosomes are contained within the nucleus. So if the nuclear membrane breaks down, it allows for the chromosomes to freely move in the cell. Then you also note that during prophase, chromosomes are going to become more visible. So when a cell is not undergoing cell division, the chromosomes are not visible, okay? But when cell division is about to take place, these chromosomes are going to coil and they'll shorten to allow them become visible. So they're going to become quite thicker so that they are able to easily be viewed. They're able to take up stains so that we can view them if we're viewing them using a microscope. So that is basically the first stage, which is prophase. Please take note of what I've said. So in prophase, centrioles move to opposite poles. Spindle fibers begin to form, nuclear membrane disintegrates, chromosomes become visible, okay? And they are becoming visible because they are coiling and shortening. I hope that is clear. The second stage that we have, our second stage of mitosis is metaphase, okay? So our second stage is metaphase. So what then happens in metaphase? So in metaphase, we have the chromosomes moving to the center of the cell. So they will align to the equator, okay? So why is that possible? So when the spindle fibers form, the chromosomes are going to be attached to these spindle fibers and they will be at the center of the cell, which is the equator, okay? So like we can see in the diagram, we have our chromosomes here. They're aligned at the center of the cell and they are attached to the spindle fibers. So these chromosomes attached to the spindle fibers via what are known as the centromeres. Remember the centromeres? We just talked about them in the introduction of chromosomes. So the centromeres, we talked about it being a structure that joins two sister chromatids. So in metaphase, we have the chromosomes lining up along the equator of the cell, which is basically the center of the cell, and they attach to the spindle fibers via the centromeres or by the centromeres, okay? We move to the next stage. So our next stage of mitosis is anaphase. In anaphase, like the diagram is showing us, we can see there's movement, isn't it? So in anaphase, we have sister chromatids getting separated from each other, and then they are pulled to opposite poles. So the pulling is such that these sister chromatids move centromere first. Remember we said in the previous, let me try and draw this for us. In the previous slide, we mentioned we have our chromosome. So let's say this is our chromosome. The chromosomes are attached to each other by the centromere, and it is at this centromere that they'll attach to a spindle fiber. So in anaphase, these two sister chromatids are going to separate from each other. They will separate. There will be a separation between them, okay? This separation is because they are getting pulled to opposite sides. So like we can see in our diagram, this is a chromatid. It's getting put to another side and it's moving centromere first. Can we see this part that I'm trying to color pink? Even the other end, the chromatid is moving centromere first. These two chromatids are what made the chromosome. And they were attached to each other by this centromere, which is getting pulled first, okay? So the question that we have is, why are these chromatids able to move to opposite poles? And why are they separating from each other? So the separation of the chromatids is as a result of microtubules contracting. So we have, remember, when we looked at prophets, we said that the centrioles are going to form spindle fibers. So the spindle fibers form these tube-like structures, which are the microtubules. So the microtubules are going to contract. So as these tubules contract, they pull the chromatids apart, okay? So each chromatid is moving to an opposite pole. That is our anaphase. So we see that in anaphase, that chromosome that was double-stranded, at the end of anaphase, it's going to become single-stranded. Remember what I told you that initially when I say it's not dividing, the chromosomes appear as single strands? So they are going to revert back to their single-stranded nature at the end of anaphase. So after anaphase, we have telophase. So telophase is our last stage of mitosis. In telophase, we have the chromatids arriving at the poles. So at anaphase, they separated. So in telophase, they're now going to arrive at the poles, and each is going to become a complete chromosome. Just as it is a single chromatid, it will be a complete chromosome. Then the spindle fibers are going to break down. Then we'll have the chromosomes becoming less visible. Remember that they became visible at prophase. So now they're going to become invisible, okay? So they'll more of like disappear. They are there just that they're not visible anymore because they're not condensed. So they're going to unwind and become thinner so that they are not visible. They won't be able to take up stain, so we won't be able to visualize them as individual chromosomes. Then the nucleolus and nuclear membrane are going to reappear. These at prophase broke down or they disintegrated, they disappeared. Now they're going to reappear. And they will reappear for each individual set of chromosomes. Remember, anaphase separated our chromosomes into two. So when the nuclear membrane reappears, it will reappear around its set of chromosomes, okay? Implying you will not have one single nuclear membrane enveloping the two, but you are going to have two separate nuclear membranes. I hope that is clear and it makes sense to what is actually happening, okay? So that is basically what happens during telophase. So telophase moves into a stage that we refer to as cytokinesis, where the cell is now going to divide into two. So the cytoplasm constricts around each as well as the cell membrane to allow these two cells to separate, okay? Other instances, the cells might remain attached to each other, like you would have, if you further study like plant cells, you know how the cells would remain attached to each other, but it's a type of cell will form in between them, but those two remain in close proximity. Other situations, you have this complete separation while this cell is able to even migrate to another. Yes. So that is telophase, which most likely is going to end in a process that we refer to as cytokinesis. So in cytokinesis, the cytoplasm is separating around itself. The cell membrane is also constricting to divide the two cells or to separate. Let me not use divide, it's just in cell division, to separate the two cells. That is our telophase. So we have looked at the stages of mitosis. What then is the importance of mitosis? So mitosis is important for growth. It's important for regeneration, for sexual reproduction, for maintaining genetic stability, as well as replacement of cells. But how is it important for these different processes? So growth. If you go back, you have covered growth and development. You define growth as being the permanent increase in size, number of cells, complexity. You had that definition. So for growth to occur, we're saying cells need to increase. And that is what we're seeing with mitosis. Mitosis is increasing the number of cells. And the cells that are forming are just like the cells that are already existing. Okay, so mitosis is important for growth. Then we're also saying that mitosis is important for regeneration. Okay. So in regeneration, we are reforming cells to regenerate. Something was there, you are reforming it. So regeneration. Then asexual reproduction. When you looked at asexual reproduction, you stated that asexual reproduction utilizes the process of mitosis, which is what we're seeing here. So in asexual, remember, it was an individual parent producing genetically identical daughter cells, and that is what we have seen with mitosis. Individual cell producing genetically identical daughter cells. Then it's also important for maintaining genetic stability. So with mitosis, whatsoever genes were in the parent are also going to be seen in the offspring. So there's that stability. There won't be any changes in the genetic composition between parents and offspring. Then we have replacement of cells. So different activities we carry out, it could be the foods that we eat, even age of cells lead to them dying, getting worn out. So these cells need to be replaced. So for that replacement, we make use of the process of mitosis. I hope that is clear on mitosis. The other type of cell division we have is meiosis. So meiosis is a special kind of cell division, in as much as it's leading to increase or multiplication of cells. The type of cell division that we see with meiosis is a form of reduction division, okay? So we have there, our meiosis is a process of cell division that produces haploid gametes, half number of chromosomes. Let me talk about this and why I said it's a reduction division. So when you look at mitosis, we said that the genetic makeup of the parent, so this parent was giving us two that are just like it. In meiosis, the offspring that are formed are not genetically identical to the parents, okay? And the number of chromosomes that they will contain is half the number of chromosomes in the parent. So we mentioned when we looked at chromosomes that we humans have got 46 chromosomes in our body cells, our somatic cells. So these 46 chromosomes are what referred to as a haploid, as a diploid number, not haploid, diploid number. Why di2, right? We said these chromosomes appear as pairs, they appear in twos. So what meiosis does is that meiosis is a reduction division, like I've said. So it's going to split this number of chromosomes. So the chromosomes won't appear as twos anymore when the new cells are formed. The new cells are only going to include a single copy of a homologous pair of chromosomes. So in our somatic cells, they were as twos, which we said were homologous pairs, right? But with meiosis, it's going to separate the homologous pairs. Okay? So the chromosomes are not going to be paired as the new cells are formed. The chromosomes are going to occur as individual chromosomes and not as pairs. So implying that when meiosis occurs, we start with a diploid number. So with meiosis, we're going to start with a diploid number. But at the end of meiosis, we're going to have a haploid number of chromosomes, implying that they're going to be divided into two. Okay, I hope we get that picture of what is happening in meiosis. So let's take a look at how meiosis occurs or what's taking place in meiosis. So meiosis has got two divisions in it. That is meiosis 1 as well as meiosis 2. In the first division, we have a reduction in the number of chromosomes that are being formed in the cell. In the second division, we have those cells making copies of themselves. So what is unique, what is different about meiosis and mitosis is something that occurs in meiosis 1. Meiosis 2 is exactly what we saw with mitosis, because whatever that the beginning cell, which is the parent cell, has is what the daughter cells will have. But with meiosis 1, what the parent cell has is not what the daughter cell will have. I hope we're getting the picture. So what do we find unique about meiosis? Okay. So the first unique feature that happens in meiosis is referred to as synapsis. So in a synapsis, or information of a synapse, we have homologous chromosomes pairing up. Remember I said that somatic cells have got homologous chromosomes. So those homologous chromosomes are going to pair up along their length. So they're going to synapse with each other. As they pair up, they're going to, I like to think of it as though they form an entangle between themselves. They entangle each other. So they form a bridge. So if you have a chromosome here, you have another chromosome here. So if this was a chromosome, so they'll synapse, huh? Though more like, yeah, that, as though they're overlapping with each other, like we can see in the diagram over there. That is what happens in synapses. That is the first unique feature. Another unique feature we have is crossing over. So in crossing over, when that synapsis occurs, the homologous chromosomes are going to exchange genetic material. So this allows for genetic variations. Remember, in mitosis, there was no genetic variation, but meiosis is going to produce cells that are genetically variable. And this genetic variation is because of crossing over taking place. So where that synapsis occurs, we're going to have crossing over where there's exchange of genetic material. Like we can see in the diagram, we've seen the two have synapsed with each other. Then they've exchanged small portions of each other. So the chromosomes that are formed each have got a portion from the other. So like this has a portion from the other one. This is also having the portion from the other one. So they've exchanged genetic material that is crossing over. And we're saying that the crossing over will lead to genetic variations. So we have our synapses, they pair up, they form a bridge, then they exchange genetic material. Another unique feature about meiosis is that it's a reduction division. So like I mentioned, with meiosis, we are reducing the number of chromosomes that are going to be present in the daughter cells. So our somatic cells have got 46, but the daughter cells formed after meiosis are each going to have 23 chromosomes. So remember that this type of cell division is something that is of importance when it comes to forming sex cells that we need for sexual reproduction. So we look at the stages of meiosis. So meiosis, like I said, is divided into two, meiosis 1 and meiosis 2. So the names are basically what we are hearing, names with regards to mitosis, prophase, and the like, but they'll have a number to them. So we have prophase 1, metaphase 1, anaphase 1, telophase 1. Then when we move to meiosis 2, we have prophase 2, metaphase 2, anaphase 2, as well as telophase 2. So the first stage we have is prophase 1. So in prophase 1, we get to see what we had seen in mitosis with disappearing, centromeres moving, spindle fibers forming. But something unique that's going to happen here is that we are going to have synapses taking place as well as crossing over. So genetic material is going to be exchanged between homologous chromosomes by their non-sister chromatids. So remember that each chromosome is made of 2 strands that we said are sister chromatids. So chromatids from 2 different chromosomes are being referred to as non-sister chromatids. Okay? So that is what is going to happen during crossing over, and something that makes prophase 1 unique, different from prophase of mitosis. Then we have metaphase 1. So in metaphase 1, homologous chromosomes are going to line up at the equator, or they line up at the center of the cell. So we note that the centromeres of the homologous chromosomes are going to be attached to the spindle fibers, okay? So you have homologous chromosomes lined up at the equator. You've seen how they are in pairs. In mitosis, metaphase of mitosis, the chromosomes were individual chromosomes. They were not paired up. But when it comes to meiosis, 1, that is metaphase 1, they're going to line up at the equator as pairs. Then we have anaphase 1. So in anaphase 1, the homologous chromosomes are separated from each other, and then they're put to opposite poles, centromeres, similar to what we had in mitosis, just that this time it's not the chromatids getting separated. It is the whole chromosome being put to an opposite pole, okay? So we note that this separation of chromosomes is something that happens randomly. So it's not predetermined that chromosomes from the maternal side, we put this side, from the paternal, we put this side. It is something that is random. So refer to that as random assortment, okay? It's random assortment of chromosomes. And it is also a contributing factor to genetic variation among organisms, okay? Then we have our telophase 1. So in our telophase 1, the whole chromosome with 2 sister chromatids on it is going to arrive at the poles. Then the spindle fibers are going to break down, they'll disappear. The chromosomes are going to unwind. Then we'll have the nucleus and the nuclear membrane reappearing. Then we'll have 2 daughter cells formed, okay? So the first meiosis will have 2 daughter cells formed. These 2 daughter cells that have been formed have got half the number of chromosomes, okay? So reduction has occurred in number of chromosomes. So this reduction is because of what happened in anaphase. In anaphase, a whole chromosome was put to an opposite pole. So meaning that you have the chromosomes that you have in the cell in 2, 2. There was 1 chromosome moves the other side, the other one moves the opposite side. So the daughter cells have got a reduced number of chromosomes compared to the parent cell. So the parent cell, say it's a somatic cell, it started with 46, like in the case of us humans, it started with 46. But as these daughter cells are formed at the end of meiosis 1, they'll each only have 23 chromosomes. I hope that is clear. Then we move into so we're saying at the end of meiosis 1, you have 2 cells, like I said. Each cell contains half the number of chromosomes, so that's half the number. one copy of each chromosome. That is for humans. Each haploid cell has 23 chromosomes. I hope that is clear. So at the end of meiosis 1, we are not going to have DNA being replicated. Remember, I stated that when cell division is starting, DNA is replicated. That's how come a chromosome has got that extra strand, which is the replicated DNA. But in between meiosis 1 and 2, there's no DNA replicated. Reasoning, when you look at the chromosomes when they're separated at anaphase 1, the chromosomes still remain with their double strand. They have not become single strands. They still have double strand. They still contain sister chromatids. Okay. So from our telophase, when the two cells are formed, we move into meiosis 2, which is exactly what we saw with mitosis. I'll still take you through, but it's exactly what you saw with mitosis. So we have prophase 2. The same processes occur. The nuclear membrane is going to break down, centrioles migrate opposite poles, spindle fibers form. The same things are happening, okay? Then we have metaphase 2. The chromosomes are lined up at the equator, the middle of the cell. So this time around, it's an individual chromosome. Remember that metaphase 1 had homologous chromosomes, but this time it's an individual chromosome. And these processes are occurring in both of the cells that you have formed from meiosis 1. Then we have anaphase 2. So in anaphase 2, now sister chromatids are separated from each other and migrate to opposite poles. They are put to the opposite poles. In anaphase 1, it was a whole chromosome being put to an opposite pole. But in anaphase 2, it is a sister chromatid. I hope we're getting those points. Then we have telophase 2. Telophase 2, you now have nuclear membranes or envelopes forming around the 4 daughter cells. Why are they 4? Meiosis 1 gave us 2. Each of these 2 that were in meiosis 1 is entering meiosis 2. So they are forming 2 more. So at the end of our telophase 2, we're going to have 4 daughter cells. And these daughter cells have got haploid number of chromosomes. I hope we're clear with that one. So at the end of our meiosis 2, we are going to have 4 haploid cells, each having only 1 copy of a chromosome. Then we're also going to, we'll have 4 haploid cells, each only has a single copy of each chromosome, okay? So we note that when you look at the cells that are formed, these are not genetically identical to the parent cell and are not genetically identical to each other, okay? Reasoning for that are processes that we have seen in meiosis, the crossing over that occurred, the random assortment of chromosomes. It allows for the cells that are formed to be genetically unique. In your brainstorming session, you were asked for why you and your sibling are not the same. Why do you appear different? Reasoning, you were formed from a cell that had undergone meiosis. So you are unique from the cell that your sibling was formed from. Are we getting that? So the cells that formed you are unique from the cells that formed your sibling. Remember that both of our sex cells, sperm cells, as well as the ovum, are both formed from meiosis. So when these 2 cells are getting formed, you are forming cells that are unique. So as they fuse with each other, they are bringing these unique features, okay? But these features are still coming from our parents. Why? The chromosomes that are in these cells are still from our parents. I hope that is clear, okay? That is our meiosis. So why then is meiosis important? So meiosis is important for formation of gametes. It's important in the formation of gametes, and it's also important for genetic variation. So let me talk about this too, formation of gametes. Gametes we know as sex cells. So they're going to be used for sexual reproduction, where a fusion needs to occur. So with meiosis, because it allows for chromosomes to be halved in number, it allows for cells that are formed from these sex cells to have half the number of chromosomes. They will have the complete number of chromosomes once they fuse with each other. The other importance we've said is its contributing factor to genetic variation, which we have said is being brought about by crossing over as well as random assortment of chromosomes. So that is basically meiosis. So we have come to the end of our lesson on cell division. So remember, we have two types of cell division, mitosis and meiosis. Please do go through the stages or the phases involved in each of these divisions and know what is happening. You can have questions that ask you to describe the stages or ask you to identify these stages, and also know the importance of the two types of cell division we have discussed. That is mitosis as well as meiosis. Okay, so thank you for listening. We shall meet next time where we shall proceed to another discussion by looking at the next topic in genetics, where we are looking at inheritance with specificity, monohybrid inheritance. So that is what we'll be looking at next time. Thank you for attention. Thank you for listening. Have a good day.